Welcome, everyone. Thank you for spending your afternoon with us. I'm very excited to introduce Cardinal, as we've titled it, A Collaborative Leap Towards Harmonization of Clinical Reporting Standards. Sounds like a lot of words, but we will get into a little bit of an explanation. So we're your instructors today. My name is Abby. I'm a developer at Roche. I work for a team called Nest, and we work on next generation tools for table creation, TLG creation, and I'm also a developer on Cardinal. I'll pass to Emily for her introduction.
Hi, I'm Emily. I work on the same team as Abby, so also working on the next gen tools for our data analysis. But yeah, I'm excited to present this to you with Abby. Back to you.
Thanks, Emily. All right, so our workshop outline for today, we'll go into a little bit about what Cardinal is. So we started about 2 years ago. We started in 2023, so there's a little bit of a history there, our journey and how we got here. We had a pivot somewhere in between, so I'd love to walk you through all of that and just to give you a glimpse of what the project looks like right now. We'll also take some time to navigate the Cardinal website. So at its current state, what can you find on the Cardinal website? What is our TLG catalog looking like? What are the templates that we have up? And also, again, because like I mentioned, we had a little bit of a pivot and we started 2 years ago, I think it would be nice to share our reflections and our learnings with you guys and our future outlook. And a call for collaboration. We would love to open the doors for more developers to join, for more people to contribute to the project. So if this sounds interesting at all to you, we would like to open that up and have you join or participate. And then we will also jump into a technical overview. So because this is a workshop, we want to get hands-on and we want to have some exercises completed. And so to give you a brief overview of the functions that we'll be using, Emily will cover a brief intro into like gtsummary and Crane and ARDs, and hopefully we'll get our hands dirty and that'll get set us up for the workshop exercises. So I just wanted to give a little or preface this a little bit. If you joined 2 years ago and this sounds a little bit familiar, it's because we were called Falcon at the time and we were presenting a package. We pivoted a little and now we're a TLG catalog or a template catalog, and we've also renamed it to Cardinal. So again, if some of this sounds familiar, you likely heard about it 2 years ago, but I'm happy to give you a little bit of the updates the projects have gone through. For today's scope, again, we want to understand and navigate the template catalog, understand the history behind it, and what you can find and how this would be resourceful for you. We'll have that brief technical overview just to get you equipped with, the functions that you would be using to help with the exercises and help create your TLGs. Workflow expectations, please ask questions at any time. Feel free to interrupt. I put this on to turn your cameras on if you're comfortable, but I don't really think this applies here. But yeah, just to have any sort of engagement, if you would like to pop in and just chat, let us know. All right, our history and motivation. So, as you know, the pharma industry has very well established standards. We have, CDISC standards, SDTM, ADAM data sets, the data types, variable names. There's a lot of standards that have been introduced, not only to make it easier to share data and to reuse the data, but we also want to make sure it fosters industry collaboration. So, these companies are using these standards to make sure that they can develop these tools as well that can be created and shared. So it would follow that, we would be able to follow this one standard and have alignment. But what about TLGs though? If we look at it, we're all creating demographics tables in different ways. We found that these differences largely can be attributed to the variety of tools that are being used to generate these tables. And so a couple of years ago, the FDA proposed an integrated guide for standard safety tables and figures. That gave us an opportunity to bring in some collaborators and work on this one tool. So instead of potentially implementing this guide individually, we wanted to work on it together. So we wanted to make sure that there was a cohesive set of TLG layouts, and we're capitalizing on this on the establishment of these standards, and making sure that we're equipped to create these tables. The benefits being that we have a common toolkit, so we're using open source or packages to create these tables. They're available on CRAN. They have many open channels for communication and making sure that feedback loop is locked in. Another thing is shared resources. So we have developers from different companies, means everyone's bringing their requirements at least to the table and we're having a discussion about it. We're able to incorporate all those different requirements. Everyone's needs are being heard when we're developing this and trying to align it with the standards. And we're making sure that everyone's happy with the outcome and everyone's aligned with the standards and the requirements. And having a single layout makes it easier as an easier entry point for collaboration. So we have more efficient data review processes as well. So like I mentioned, we had a little bit of a pivot. So if you were introduced to Falcon, it was a package that was designed to create functions that would generate the FDA standard tables. And you also had the option of choosing which table engine that you were using. So it was between tplyr, R tables, or GT and gtsummary. Around the same time in early 2024, CDISC also published an analysis results dataset standard. So this was a structured way to store your analytical results. And we also saw a push for retaining these raw data sets, not only for QCing and data review and meta-analyses, but also to make sure that you can potentially create a table afterwards using these stored data sets so you don't have to keep using your raw data to create the tables. So in addition, we were finding it at the time, we were finding the effort to accommodate the different table engines in one package. It was making our functions look very clunky, and the benefits also seemed marginal. So what we were doing was centralizing the traffic to this one package and saying, if you want to build, FDA table 2, you can use it with these 3 functions. But it wasn't really minimizing the amount of work that would be done to help, or maximizing or capitalizing on the fact that we have these standards available. So being still in the early stages of development, we asked, like, can we afford to be proactive and address this in addition to creating the templates that we already had made? So again, rather than developing Falcon to accommodate the different table engines, can we use a singular package? So having researched those 3 different packages, or having interacted with them when we were developing Falcon, gtsummary really stood out. So for us, the benefits was because it was widely adopted and had a large user base, it had more resources. So if you were to look up a question on Stack Overflow, or if you wanted to look at any of the GitHub issues that have been closed or any of the PRs, and the code was openly accessible. In addition, it's also been presented in many conferences like POSIT, R in Pharma. So more people knew about it, and therefore we knew there were more resources available to help. We also had an open channel of communication. So Daniel Schoberg, who's the author of the package, he works at Genentech, and he's also part of the NEST team. So we were able to kind of shorten that feedback loop. So if we had any questions, he was able to address them, or the other way around. So again, that made the development process very quick and efficient. Another thing is readable code. So, line by line, it was a lot easier to understand gtsummary and get onboarded onto that. Again, having worked with the 3 different packages, it really stood out. Another thing, that complex tables were easier to create using simple tables. So a lot of the packages, the structure made it difficult to add these complexities, or add, one-off, or change the aesthetics of things. But gtsummary is actually meant, or it's encouraged, that you're building these smaller tables and cobbling them either like through merges or stacks to ensure that you're able to get exactly the table that you want in their component pieces, which was like a dogma, I guess, that we really appreciated. And then recently, it was also refactored to have an ARD backbone. So like we mentioned, with this new standard being introduced, and that being the way we wanted to pivot, this was very conducive to making sure that we had all of the information available and we could build off of that. So we were already pointing in the direction that the industry was going. It was also compatible to cards and CardX. So these packages are being actively developed to facilitate, the ARD creation step of it. Also, gtsummary can hold on its own. It's a very well-known package. It has over 1 million installations from CRAN. It's very widely used. It's won many awards, and it has a lot of contributors. So it was a very clear and obvious choice to use for Cardinal as we made this pivot. Now we introduce Cardinal, an industry collaborative effort with the aspiration of open sourcing a catalog of harmonized TLGs for clinical study reporting, and simplifying the processes for output review, reuse, and meta-analyses. Essentially, we wanted like a one-stop shop, some place that can be the industry's TLG catalog. By incorporating ARDs, we're ensuring that that standard is being adhered to, and we have a way to capture that and retain that information in addition to creating those tables. This is a little aside here, but if at any point you can guess why we chose the name Cardinal, you can drop it in the chat. Okay, so our current progress. So we talked a lot about collaboration. How exactly does that work? Our project coordination is kind of like in 2 sections. Our product owners, so having awareness of the data standards, where the industry is going. Are there any changes in the outlook? They're able to kind of set the tone and set the project roadmap. And using that, they're able to kind of refine the requirements and prioritize the different templates. So right now we're working on the FDA guide, but we're also able to incorporate more tables from the different companies individually. And then that works our way over to our developers. So right now we have 7 active developers from those 4 different companies. We work on an agile development kind of framework. So we have those biweekly stand-ups. We're able to post issues on our GitHub project board, and we're able to track progress that way. Everyone's able to collaborate through, pull request reviews. So we're all kind of aligning on the same standards, and we're able to introduce that consistency because, again, everyone is working towards making that one FDA guide, following the data setup, following the data manipulation, following the table creation. Is it easier to do ARDs first or tables first? So we're all kind of combined in that one section, and we're all developing together. So our journey so far, again, this started 2023, early 2022. We had a little bit of a kickoff, and then we had our team established. So we had a group of developers joining, we had our product owners joining, we had a lot of the housekeeping things set up. So for example, the stand-ups that we're doing bi-weekly. And so we had that constant communication. Throughout 2023 is when we did the active development. But again, at the time, it was more set up as a package. And as Falcon is when we approached, increasing awareness by attending these different conferences like RN Pharma and Fuse EU. And then in 2024 is when we did the catalog pivot. So again, instead of reinventing the wheel and creating yet another package that can create a table, we were pivoting to the catalog where we have these templates for the FDA integrated guide in addition to other tables that might be specific to your company or for your studies, and that might be a useful resource for other people, and we wanted to include that here. Going forward in our future outlook, we would like to work closely and more in alignment with CDISC ARD. So now that we have the backbone for it, we'd like to be integrated into the catalog. We'd like to expand that a little bit further. So now if we were to do a deeper look into our actual website, what's entailed in our catalog. This link we can put in the chat as well. It would be great for you to poke around and navigate on your own time if you'd like to. It's updated very frequently and it's a great resource. So in addition to the catalog, we have other things like our past presentations, the slides are available and our resources, the FDA guide is available in the resources. If you'd like to join as a collaborator or join as a developer, we have all the instructions available there. So it's a great resource. So this is our current template catalog. It scrolls down further. So it looks like we have roughly 27 templates currently. And so all of them are organized by their different categories. So even if you don't find the table that is specific to your study or what you're looking for, you might be able to find something that's adjacent and use that as a template for the table that you're trying to create. And within each catalog entry, there's 4 components. So one being the table preview, so what it would look like. The next part is the data setup. So we're trying to showcase how you can manipulate the data to feed into your gtsummary functions to create the table that you would like. And the third component is building the table. So this is where we introduce the gtsummary functions. Some of the tables, if it's specific to that and it's required, we can have manipulations of like the footnotes, the headers, any of the labels if they apply to you. So this is just kind of showcasing how you can change the aesthetics of your tables if needed. And then there's also a section on how to extract the ARD. So gtsummary has a function called gather ARD. And once you've built the table, you're able to pull the ARD object. And that is something for you to retain every time you build the table. Again, since we've worked on this for a couple of years, we've had a bit of a reflection. I think it would be nice to share our learnings with you guys as well. The key learning is collaboration entry point is significantly lower when an industry-wide standard is established. Even the FDA implementation guide, we're ensuring that there's alignment in what we're building and what we're incorporating. So there's consistency in how we're approaching the data setup, there's consistency in how we're approaching the table creation, the ARD creation. In some instances, is it better to create the ARD first? And because we're having these discussions as a whole between all of our developers, there's alignment and consistency among us, and we're able to propagate that in our individual companies as well. Developers are also motivated to work on an open source project, which means new career opportunities. I think having these standards, the benefit is that we're able to develop these tools that are going to become very widespread, and you're gaining expertise and able to contribute to things that are going to make an impact in the industry that's greater probably than your role within your company alone. And building an open source resource together across pharma companies is less resource intensive. So even if we were to look at the FDA guide, having 2 to 3 developers potentially implementing it in one company alone versus 7 people who are working on the same number of tables. And we're having these discussions proactively about, how can we approach making the alignment consistent and making sure that everything looks correct and everyone is approaching it the same way and utilizing the functions to the best of its abilities. So I think that's making it a lot more efficient. For our future, we want to, again, of course, engage more companies and want more collaboration. So again, we'd like to introduce and invite more people to join if they have more tables that they'd like to contribute outside of the FDA guide. If there's things specific to your company that you would like to contribute and you think it would be a helpful resource, we would love to invite that. Another thing is an industry harmonized TLG standard would replace all internal standards and the implementation is freely accessible. So having these tools that are open source and publicly available and having all these people contribute to the same project will make sure that everyone's kind of going towards the same goal and they wouldn't need their specific internal standards to adhere to. Call for, again, we'd like to invite more people to collaborate. So the best time to join was a year ago, but the second best time is now. You can find us or our information on the Farmers website because it is a package hosted under that. And then we also have a Slack channel available with all our developers. So if you would like to join and get a sense for, the community that we have currently going on, that's available. And then our website itself, the Cardinal website, is a very good resource, and we will share that link after this as well. Before we move on to the technical aspect, this is just a brief acknowledgement for all the different developers we've had. We had some rotations in between, but this wouldn't have been possible without all the people here. All right, so I will pass over to Emily for our technical overview of our functions, and then we'll move on to our exercises.
Thanks, Abby. Let me just pull up my slides. Okay, so I'm just going to go over a couple of the functions that we use from gtsummary and Crane for our table development. So this is just a couple functions that we use for like a lot of the basic tables. You can sort of build off of these functions a lot to build out the catalog. Of course, we have like several tables that are more specific that use like different, more custom functions, but these ones can get you a lot of tables done with very minimal effort. So we're just going to cover some of these. And I'm going to start with TBL summary, which is just taken from gtsummary. So for the purposes of this presentation, we're just going to use some data from the Pharmaverse Atom package. I just reduced the size of the ADSL and ADAE datasets, so just made them shorter for the purpose of the example. And then we also added a variable called female, which is just a dichotomous variable indicating whether the sex is female. And that's just for the presentation purpose as well. So this is just sort of the basic out of the box TBL summary. So you just include the variables that you want to use in your table, and then you just get this as the default layout. So we have our age variable, our ethnicity, and our female. These have default types. So age would be continuous, ethnicity is a categorical variable, and female is dichotomous. And the default statistics are listed here. So we have median and interquartile range for the categorical, or sorry, the continuous. And then for categorical, we have N, which is just the count of subjects with this specific value. And then we have percentage, which is the N value over the overall N. So that would be this number here. And that's just what we get here. We have the same statistic for dichotomous. So dichotomous would be any sort of logical variables. So if it's coded as 0, 1, true, false, yes, no, you will get the same sort of one-line summary indicating how many yeses you've got. And then the labels are just taken directly from your data set as well. So that's just these three right here. And then the labels for each of the levels as well. It's very easy to make customizations with gtsummary. So a lot of functions use a sort of shared syntax, but just to look at TBL summary, we can use the by argument just to stratify column-wise by a treatment variable. So we have arm2 here, and that will just split, again, our output by treatment. We have type, so here we're just changing the type of the age variable to continuous 2, and this will just change our age variable from a one-liner. So we previously had median and interquartile range right here on the same line as the label, and continuous 2 will just make that so that it is on separate lines. So you can have multiple statistics listed instead of just one single line of statistics. And you can see that here we just specified the statistics. So here we now have mean standard deviation as our first statistic, and then we have min max as our second, and these are printed on two separate lines. And then we just changed our female variable to have a percentage, and then also have the fraction in front of that. So we have a more like explicit, I guess, version of what we had above. And that's just taking this n value from above and pasting it down below. And then we can change the labels pretty easily as well. Here we just change age to age years, and that's just changed directly in the table as we specify. And digits is used to format the statistics that you get out. So here we're just changing the number of decimal places for the SD statistic to 1. So now we just have 0.6, 0.1 instead of what we had before. You can also use functions under digits. So if you have a more complex sort of formatting that you want to use, you can do that as well. But it's pretty easy to use and there's a lot of different options. So that's what we've used there. So gtsummary is pretty easygoing in terms of what you can use as input. We have a formula syntax, so you can put your variables and then use a little tilde and then specify the instructions or value afterward. You can also just use a list, so you could do, for example, age equal to patient age instead of the tilde. And then if you have more than one set of instructions that you want to give, so for example, here we're changing the label for two variables, you can just do a list and then again use either the tilde, the formula syntax, or just use the equals and just have it as a list like that. So it's pretty sort of customizable. You can use whichever is easier depending on your input. And we have a bunch of helper functions as well that can help with specifying your formulas. So we have all categorical, all continuous, which you can use to, for example, change labels, or you might want to change all of the statistics for a continuous variable to mean instead of SD, and you can do that pretty easily. You can also use all stat calls to make modifications to all of the columns if you're using a by variable. That's pretty useful as well. And then we have a bunch of different add-on functions that are applicable to most gtsummary functions, including TBL summary. So there's the add functions, which will add an additional column of statistics or information such as p-values, treatment differences, overall statistics. And then we have the modify family of functions, which will help you format your table. So you can change the bolding, you can add headers, you can change labels, footnotes, etc. Here's an example of one of the add functions. So we have add overall, which just adds a column of overall statistics. So that's what we get here. And that's just taking all the values from all of the previous columns and summarizing it in one right there. We can also do add n for non-missing counts. Add p will add p-values. Add difference will add mean and rate differences between groups. Just to take a quick look at some of the modify functions, you can use modify header, which will just change the header values. So up here we've changed our previous labels for our treatments to group A and group B. We also added a spanning header, so we're specifying that we want it over all of these stat columns, and we're putting drug there. So that's what we have here over both of our stat columns. Here we have modified footnote. So we just changed our footnote right here to this text, and we're using that again for all stat columns. So we have this little superscript here that indicates that for each of the stat columns, that footnote applies. We can use the show header names function just to see some more information about our headers. So that would be sort of labels. You can see here the labels, the levels before you've changed labels, if you've done that. We have sort of the raw statistics. So we have big N, small n, and percentage. And again, here we have all stat calls. Will show you that anything that's labeled with stat underscore something will be selected when you use all stat calls. So here, just an example, you can use add stat. That will just add on whatever statistic you specify. So there's a lot of customizability with that one as well. And you can use a single column or you could do multiple. So you never have to like lock in everything right at the beginning. Once you're making your table, you can always add in with subsequent calls to other functions. And we have a lot of documentation on all these functions. So again, there's just the reference page for gtsummary. You can check that out. Or there's a more detailed summary vignette for the TBL summary function specifically, and that's also available on the gtsummary website under articles. I'll talk briefly about TBL hierarchical. This is also a function in gtsummary. So this is used to make hierarchy tables. That's sort of what we've coined for these tables that are indented according to different variables. So you start the list of variables you want to include with the uppermost level. So that would be SOC right here. And then we have AED code. We have that as our second variable right there. So it sort of creates a hierarchy structure where everything is nested within the previous hierarchy variable. And you can do more than two, but just for the purpose of this table, we've included two different levels. So this will, since we're using TBL hierarchical, we can specify an ID variable, and that will allow us to only get the counts per subject or participant. So for example, if one participant experiences a single SOC multiple times, they'll just be counted once here. If you want just the plain counts, so disregarding the IDs, you can use TBL hierarchical count, and that will give you that. But for our purposes, we are using just the plain TBL hierarchical with the ID. So this would indicate to us that 4 subjects have experienced a cardiac disorder, 2 have experienced an atrial flutter, and then these percentages are again just this number here divided by the big N value. So that's used in a lot of our tables within Cardinal, and it's pretty useful. And you can also use a sorting function and a filtering function, which we'll get to in the exercises on these tables to sort of customize your output as well. I will cover TBL merge and TBL stack next. So those are just some basic functions that are used to sort of combine your various gtsummary tables if you have a specific layout you need, if you need statistics from multiple summaries all in one table. So we'll get into that next. So TBL merge is specifically for side-by-side tables. So if you want everything sort of next to each other, here we have TBL-N, which just adds a table with a column for N values. And then we have a separate table called TBL-Age, and that is where these two columns come from. Then we add them together with TBL-Merge. So you can see that call right here. We just put in a list of the tables we want to include. And assuming that you have similar structures, so the same sort of rows in both tables, it will combine like this. So everything's sort of merged together into one single table. You can also stack tables vertically. So we use TBL stack for that, very similar to TBL merge, but instead of side by side, we want things on top of each other. And again, this function would sort of expect that you have similar structure. So maybe like only one column here and then the label column in order to stack everything sort of cleanly. So we have a table with drug A, which is our placebo, and then here's drug B, which is this one right below. And we're just stacking those right on top of each other. And then we use the modify header function to change our header to summary statistics so that it sort of describes both tables. The default would otherwise just be to take the header from the first table. So we want to sort of make it more generalized to both. And again, there's many ways to sort of cobble tables together. You can add like the post-processing functions like add stat to add things in. After the fact, you can use TBL merge, TBL cross, TBL stack, just to sort of stick things in whatever direction you need, put summaries together from different data sets, that sort of thing. And then it sort of makes it all look more cohesive. So it's very often that we do this. So just to cover the ARD side of everything, ARDs are sort of the backbone of what we use in gtsummary. So every time a gtsummary table is created and calculated, everything is first created as ARDs, and then the gtsummary functions internally will take those ARDs and translate them into a table format. And then that ARD object is also saved from every calculation. So this is just a basic example of a TBL summary call. And then we can use gather ARD from the gtsummary the gtsummary package. And that will just extract that ARD object. It includes, for example, like all the stat values, the labels, the stat names without labels, the variables, and then sort of the by variables that you used in your table. And ARDs are very useful for QCing. So anytime you need to sort of compare two tables or you need to check your values are correct, an ARD is going to be easier to use than a table because you can sort of see the raw formats and things like that. You can take an ARD out of a table after the fact, so you don't have to like pre-plan. You can also start with an ARD and then just put that into a function that will create the table from that. So you have a lot of different options if you're working with ARDs. Here we can look at a basic demographics ARD. So we're just using ARD summary, which is a basic function from cards. That's our ARD package that we use sort of in the background. And we're just going to take these three summary statistics. So that's what we're expecting our ARD to contain, and we're using this as our by variable. It's pretty similar to gtsummary, the syntax, but just a bit different. And then we can use ARD stack. So you can combine sort of multiple ARDs into one with that function. We'll take a look at the ARD first tables. So these are tables that are created from an ARD rather than tables that are created and then an ARD is extracted. So these are our gtsummary basic table functions right here. These are just four of them. And then these are their equivalent ARD first functions. So if you put an ARD into one of these, you should get a similar output to what you'd get with the TBL summary function. Here is an example of some ARDs that are stacked. So again, we have the ARD summary for our continuous variable age. We use ARD tabulate for the categorical variable ethnic, and then we have ARD tabulate value for our dichotomous variable female again. So we're combining all these into one single ARD with ARD stack. And then we use the attributes and missing arguments. We set these to true. These will just make things look better. They sort of will encapsulate the labels and different attributes from the data frame and bring them into your ARD so that your table will look better with the default values. And this is what happens when we feed that ARD that we just created into TBL ARD summary. So this is the table we get. It's just displaying all of those statistics we calculated in the ARD. As default, we get these as the statistics, but again, you can use the statistic argument here to customize the layout and change things like that. And we are going to talk about Crane next. So Crane is sort of an extension to the gtsummary package. We created this at Roche to sort of add some thin wrapper functions in for the gtsummary functions. And these sort of help us to apply like the defaults that we'd use at Roche for aesthetic purposes. So like, for example, we like to have our statistics on multiple lines instead of one by default. So we use continuous 2 as a default type instead of continuous and things like that. But these functions in the Crane package help to produce some standard hierarchical and survival analysis tables, as well as listings at the moment. So we can cover some of those. We have TBL baseline change. So that's used to create a analysis value and change from baseline table per visit. We have TBL hierarchical rate and count and rate by grade as sort of extensions to TBL hierarchical. So these functions will format rates and counts and rates by highest toxicity grade, respectively, sort of without having to do all the extra processing. So those are sort of helper functions when you need to create those specific tables. And then we have a TBL listings function as well that we use often to create listing outputs. So that will sort of just format your data frames a bit more nicely for output purposes. This is one of our main like wrapper functions in Crane. So we have TBL Roche summary instead of TBL summary. And again, as I said before, we just like to have our statistics on separate lines like this. And these are default statistics rather than what we had before. We also have a n value here. We call that the non-missing value instead of the missing value, because that's what we typically like to show at Roche. But just general things like that are included in these Crane functions. So you can use this is just the data setup for our next example, I guess. But if you're creating a lab table, you might want to create a table with visit values and then change from baseline values. And instead of having to use TBL merge with the eval and change variables, we can just use one simple call to TBL baseline change from Crane, and that will just automatically apply all of that for you. So this would be the output of that function call. This is just the default out of the box output. So we have our baseline visit. It knows to leave out the change from baseline values here. It will automatically format with these statistics, and it will separate everything by visit like this. So that is also commonly used in some of our outputs, and we have a couple of tables in Cardinal as well that use this function. Just to break it down a bit, we have this section that's from TBL summary for eval, and then we can do the same thing with change, and that's where these columns come from. And then we just use TBL merge to put everything together and sort of order these variables as we like. So that is where that comes from, but you just need a single function call instead of needing to call all of those. And now we're going to get our hands dirty. So I'll pass back to Abby to start us off with some exercises using these functions. And we are going to be working in Posit Cloud. So we have a couple of exercises set up for you guys to try out. And hopefully that goes well. Abby, do you want to take over?
Yeah, thanks. So does everyone have the link to the Posit Cloud space? Before we move on to the exercise, were there any questions based on what we talked about just now or any of the technical details? There were some questions that came up in the chat. I tried to answer or at least provide a resource that you can do some further reading on. But yeah, if there's anything that you'd like to address before we get into the exercises, let me know. Um, okay, was everyone able to access this? Are there any issues with the deposit cloud space? Can you elaborate on the denominator equals ADSL when summarizing the AE table? Emily?
Yeah, I can cover that question. So when we're making that AE table, sometimes you want to use a different data set as your denominator values. So you take those n values in the header from a different data set than, for example, the ADAE data set that we use for our AE table. So when we set denominator equal to ADSL, it will just take the counts for each treatment from that data set instead, and those will be used for the denominators when we calculate the percentages for each of the SOCs or AEs in that example table we had.
All right, okay. So keep the questions coming. I like this. I think this is how we learn. But yeah, as we go through these examples, I think it'll help. So what I'm going to do is I would like to direct everyone's attention to the demo 01 demographics table. So I'll give you guys 5 minutes to get started. And I think we've supplied a lot of resources where this should not take a lot of work. But yeah, we'll give you 5 minutes to get a head start. So we've divided the script into the three sections that you would have found in a template catalog entry. So that would be your data pre-processing or the data setup, building the table, and building the ART. So we have some hints in the script here, but we would like to give everyone a chance to kind of put their knowledge into practice, what we just talked about. And so I will give until, so right now on my clock, the time is 3:46. We'll go until 51 and I'll check back in with you and then we can get started on the solution.
Hey, Abby, are you sharing your screen now by chance?
Yes.
Okay, cool. Up on the top left, do you mind closing out the left-hand side on yeah, over there. Yep, close that out. It's going to make it nice big. And then at the top, there's a message. You might close that out too. Just above it, just above that, that little blue X. Yeah. And then might zoom in just a tiny bit. There was a comment in the chat box just saying if you could zoom in just a little bit would be helpful. So you might see if yeah, that's more than enough. That'd be perfect. Thanks so much.
Okay,
Yep.
no problem. Okay. Well, we do have three tables we want to walk through, so maybe I'll go do this step by step for this first table, and then I'll give more time for the second one. How does that sound? Yeah. Okay. So, actually, let me pull up the table we're trying to build so everyone has a good understanding. So we are looking at this demographics table. I'm sure all of you have tried to create this at one point because it's very popular and everyone builds it. So we want to have these characteristics. This is what we want the table to end up looking like. Categorical variables and our continuous variables defined with these statistics and having them ordered this way. Okay, so if that's clear. So going back to here for our data pre-processing, because this is a safety table, we want to filter for observations with a safety flag with as yes. And if you did a kind of prelim exploring of the data set, you would know that there's some screen failures in the treatment variable, which we don't want to include in our table. So we would filter that out. And then to build the actual table, so again, TBL summary makes it very easy. You can just pipe the data set into the function itself. You don't need to do a lot of extravagant like filtering at that point. The include argument is mentioning which variables you'd like to include in your data analysis. So when we look at our mockup over here, we want to include the sex variable, age group, ethnicity, race, and these are optional, which we haven't included just yet. But if you wanted to include a continuous biomarker or categorical biomarker, whatever applies to your actual demographics table, you can add that in here. The second component is our by. So we want to stratify by our treatment as we see over here. So dividing it by our treatment variable. And so we would specify that using the by argument, as Emily mentioned during her technical walkthrough. Then we want to specify that for our continuous variable, so in this case age, our results, or sorry, our statistics span more than one line. So we need to specify that, saying that for every continuous variable, we want it to be of the continuous two type. That one is right here. Then for We want to also specify the statistics that we actually want visible. So for all of our continuous variable, we want to see the mean, the standard deviation, the median, the minimum, and the maximum, and formatted this way. So you might have recognized this as glue syntax. So this makes it very easy, again, to know how it's going to appear in your final table and also how to supply it to this function for what statistics you want. In the same way, so for our categorical one, we want the number of observations, or n, and we want the percentage beside it in brackets, which is how we've arranged it here. We also want the age group because so by default, the columns or the labels that you will get is the actual variable name. And sometimes that's not human readable or very pretty to see. So you can update the label using the label argument. And finally, if we look at the mockup, the last thing we need is the total population column. So we want an overall column. And if we ran the table as is without the overall, it wouldn't include that because we haven't specified it. So if we look at the table, it has all the characteristics that we were looking for except that add overall. So like Emily mentioned, we have add functions and modify functions. So this last one right here is just to add the overall. And then finally, our table would look like this. And now we've created FDA table 2. If we wanted to now gather the ARG from this table, we would use the gather ARG function. And our resulting ARG would be this data frame that has all the statistics as. So there would be one specific for the overall column, and then another one for our treatment columns before we added the overall. So this would contain all those statistics. Are there any questions or anything confusing, anything you'd like to go over? Okay, nice. So I think, where is ADSL saved? How can missing categories be handled? So there are arguments within TBL summary that lets you know how to go about handling unknowns or missing observations. You might have noticed in the setup that there was something called a theme gtsummary compact that kind of predefines what you would see in terms of like the missing or how it's going to be how the missing values are going to be handled. So if you would like to specify that or customize that a little bit, there are arguments in there that we can provide for you. Can you add a custom footnote? Yes, there is an add footnote function. I'll pass over to Emily to start on her second exercise, and I will add all these answers in the chat. Can you export these tables into Excel? I believe you can.
Okay, I'll just pull up the second exercise. So you should be able to find this the same place you found the first one. So this one's just demo 02, and this is going to be an adverse events table. So this is all these tables that we're using for our exercises are in Cardinal already, but this is just some guidelines and examples on how we created those. We might have done a bit more formatting in the actual catalog, but just for the purpose of these examples, we have a basic version of each one. So this one is going to be similar to the one that we saw in the slides before. So we're going to do an adverse events table by system organ class and preferred term. I've already got the exercise up here, but I will give you guys 10 minutes to work on this. So it's 4 o'clock. I'll give you till 4:10 just to try this out yourself. It should say which data sets you need, just those two right there. Some of the functions you'll need are specified as well. And then we have a couple of extra processing steps you can test out as well. We didn't really go into these functions, sort hierarchical and filter hierarchical, but I will demo those if you aren't able to get them working once I go over the solution. So at 4:10, I will come back with the solution. Okay, hopefully that was enough time for you guys to make a start on this table. So I'm just going to go through the step-by-step solution for this one now. So switching over to that, we have the data pre-processing. So first we're going to just filter our ADAE dataset from Farmer versus Adam on the SAFFL safety population flag. So we're just going to take the safety population since that's what's specified in the table name. So we just run that. And then we're going to build the table. So this is just the basic structure. We're going to start with a call to TVL hierarchical, and we're just piping in our ADAE data set there. So you could also just say data is equal to ADAE if you're not into using pipes. And then we select our hierarchy variables. So here we're just going to use two, the AE bod sys, so the system organ class variable, and then AE decode, which is our preferred term variable. And we are going to stratify our whole table by the treatment variable. So that's what we have there. So we want each treatment to be its own column. Like we talked about in the presentation earlier, we are going to set an ID column. So we want to set that to usage ID. So for each unique patient ID or subject ID, we're only going to count each incidence of these variable values once. So each SOC will have a count for the number of times a patient got that SOC, not the number of times the SOC occurred in total. And then here again, we're going to put a denominator. We're going to use the ADSL dataset. So we're going to want to use ADSL so that the entire population is represented here. So you may have some patients in ADSL that are not included in ADAE, but we still want to include them in the totals. That's where this denominator might come in. And just to run the basic table, we will run that before we do the sorting and the filtering. And this is what we get. So this is our entire data set. It's like pretty big. If you look through the whole thing, we have a lot of SOCs, a lot of AEs represented here. So just to look at the first one, we have cardiac disorders. Here's the N values. So there are, for example, 16 subjects with this treatment that experienced a cardiac disorder or one or more cardiac disorders. So that's where this number comes from. And then the 17% will just be this 16 over the overall N from ADSL. So that's the 96. That's where you get those numbers from. And then same thing for all the other numbers in the table. Here we're going to use sort hierarchical. So the instructions said to try sorting the table. By default, a table will be sorted in alphabetical order. But if you use sort hierarchical, the default value for that is to sort by descending frequency. So if we just leave nothing here, the default will bring the SOC with the highest counts to the top. So that's what we get here. So you can see this has been changed. And then we have a filter function, filter hierarchical. We're going to set a filter so that every row sums up to at least 5 in the N values. So for example, here we'd have 3 plus 14 plus 13. Since we're using the overall suffix here, it's going to tally those up. So get the overall N value. So we can just apply that here and our data set will hopefully narrow down and our table will be much shorter. So here our table is much shorter. Now you can see it's just showing the AEs with the highest counts. So everything here will sum up to at least 5. We have 1, 5, 5, so that's 11. Here we've got 5, 1, 0, so that's 6. So yeah, all of the lower values are still greater than 5, so that's what we wanted. And this is our finished table. Now we might want to gather the ARD from that. So we're going to use gather ARD, just our basic gtsummary function to extract the ARD. And then we can take a look at what comes out there. So let me just run that again so it's wider. So here we have sort of the overall values. So it's going to give you all of the information from the header as well. So here we just have the treatment variable information from up here. And then following that, you'll have all of the statistics for each line as well. So that will encapsulate everything. We also have a function TBL ARD hierarchical. So if you were to make an ARD for a hierarchy table, you can just put that directly into TBL ARD hierarchical, and it will give you a hierarchy table formatted like this. So you can go either way, ARD first or table first for this kind of table as well. So Abby, did you want to go over the last example, and I can check if there's any questions for this one?
Sure. Okay, so we're going to try and create a lab test results table. This uses the change from baseline function we briefly mentioned from the Crane package, but it is loaded in your environment, so feel free to look up some of the documentation if you'd like to look into it further. But essentially, we're trying to create this table, not exactly this one because this has multiple parameters. We would like for you to do the analysis value or value at visit and change from baseline for one of the parameters. So this is what it would look like. And if you open up this script, the Roche table LBTO1, oh, this is not, we would like you to fill this part out. Again, there are some notes here to help you with your filtering and the steps that you would have to take. So we'll give you another 10 minutes to work on this one, and we'll reconvene and go over the solution for this one. All right, so like I mentioned, we're trying to create this lab test results and change from baseline by visit table. So your mockup is going to look a little bit different from this just because this is stratified also by different parameters, but we're only solving for one. So here in the data pre-processing, we're using the ADLB or lab results data set. Again, because it's a safety table, we're filtering for the safety population. If you do a screen of the ADLB data set, you'll notice that some of the analysis visit numbers are like 9,000, and that's just to show like invalid visits. So if it's something like pre-baseline or post-baseline, things that don't really apply to your analysis, they would be coded differently. So to filter these out, we are asking for visit numbers that are less than 9,000. And then again, we want the visit to be this is just to condense it. So if you wanted to add more of the visits, you can omit this filtering step, but we're keeping only the baseline and week 4 visits. And we wanted this for one parameter, so we're selecting albumin here. So if we run this, we have our data set that we're now going to pass to Crane's change from baseline function. So if you were to, this is possible to do using functions in gtsummary such as TBL summary, but you would just have to call it multiple times and then perform a merge. So like Emily had mentioned in the technical overview part, this function was created in Crane in order to do all of those steps within this one function call. So here, all we're specifying is which exactly the baseline level is. So here, this is to make sure that in the table, the change from baseline part isn't calculated for the actual baseline visit. We're trying to figure out which one the baseline like reference is. And we're saying, again, we're going to stratify by treatment, and our denominator is our subject level population. So for these percentages, if they apply, it would be for the ADSL header counts. So if we run this, we should be able to produce our resultant table, which is this right here. There we go. This is clearly invisible. I think I haven't changed the font size or anything, but yeah. So we created this duty table. And for the ARD, again, simple instructions, we gather the ARD, and this will show you, or this will produce the ARD, and you can also save this to an object if you would like to save it, save this data frame instead of just printing to console. Are there any questions about this last example or this function? Lab results table, how do we specify? Okay. Emily has answered that question. I guess I can read it out loud. In the lab results table, how do we specify the decimals for mean, median, as standard deviation based on the number of decimals in the raw data? Emily has responded that we are working on adding a function to customize the decimal places according to the raw data precision. Okay, cool. Can you explain high-level overview on ARD? So I think we covered this earlier in the presentation. So when we include the slides, I think that would be helpful. And then I can also provide some like material or resources on where we're getting the ARD kind of standards from. Is that helpful? Okay. Just for now, before we close off or anything, I just wanted to show you the Cardinal website so we can help navigate. So you might have noticed that I was clicking onto this very frequently, and this is what we kind of use to develop those exercises or to extract the code for the exercises. So if you went into Cardinal, this is on the landing page. We have a brief overview, our different contributors, who's part of, which team. And then we also have some upcoming talks and presentations. So all of this information can be found on our homepage. We also have these buttons here that we can help navigate to different sections. So for example, if you wanted to know about our collaboration, and this has some steps on like how to join as a collaborator. So if you would like to join as a developer, these are some reading materials and things to keep us up to date. And then we also want to bring attention to the resources tab because a lot of people had asked for today's slides. So we haven't posted it yet. We will post it right after this presentation here, but you will find it under this tab over here under resources. We will put it under R Pharma for 2025's workshop. We also have our different about section. Again, this kind of redirects to the same page as the collaboration, but the bread and butter of our of this website is the template catalog. So I showed screenshots of this earlier, but this is essentially where you'd be going. So we're hoping that this will extend beyond just the FDA table. So if you'd like to contribute an FDA table, feel free. But you'll notice that we also have some company-specific ones. So that lab one was specific to Roche, Roche LBT01. So if you have templates that are specific to your company, again, that you think would be a great resource, that would be nice to add in here. And we would just add the tag for your specific company so it's easy to find. And as I mentioned, these were screenshots earlier, but this is just nice to demo it live. For each of the table, we'll have the table preview, so what exactly your code is going to generate, the data setup, how to build the table, and it'll show you again the resulting table at the bottom of this, as well as the ARD, so once you gather it. For some other templates, if they are very complex or they benefit from starting from an ARD first approach, these tabs might be flipped over. So you will build the ARD first and then you would create the table. But all of it would be very clear and apparent on the website. And then the one last thing that I wanted to bring your attention to is we had a lot of questions about gtsummary and their functions. It's good to note that gtsummary, or Daniel will actually be presenting at R Pharma as a talk on Wednesday. So I can provide, I don't think the link is up, or I will try to find the link to put on there. But yeah, if you have questions or you're very interested about gtsummary and have more questions, related to that, that would be a great talk to attend. And I believe it's on Wednesday. Okay, I will find it. I will send it. But yeah, are there any questions? Emily, is there anything you wanted to include or talk about?
I think you covered everything. If there's any questions, we can stick around and address those. So if you have anything to ask, or if you think of anything later, you can always post to our GitHub, which is accessible from the website as well.
Okay, so yeah, up here, these little widgets, I guess they can help navigate to the different pages. So this will take you to our GitHub repository. And this has all our code openly available. And if you have any issues that you would like to create, or any templates that you would like us to add, feel free to create an issue here and we would be able to address it or put it on our board for our developers to work on and pick up. Are the tables for parallel studies or fit for crossover trials? I don't think we've encountered any so far, just because we were following the FDA guide templates. But if there is a table that you would be using for your own studies and you think it would be helpful, I'm sure it's possible with gtsummary because, like we mentioned, complex tables can be built using simpler tables and kind of merged together. So if you come across or if you find it helpful to create a table for yourself and you would like to contribute that to the catalog, that would be a great way to add it. But to answer your question, yes, it should be possible with gtsummary functions. Yeah, I guess there were some questions that came in, like technical things on how to add missing. We just weren't prepared for it because our data doesn't have like missing values and such. So what I can do is add to the script at the bottom and say like these are some common questions that were asked, and I can display the code for that after we've manipulated the data a little bit. So if that'll be helpful for you, feel free to check back into like the assignment portal of Posit Cloud, and you should be able to find the updated script there.
Yeah, that's a fantastic idea. You also mentioned you're going to put it on the Pharmaverse page. You could have a small section there too, I suspect, which would be good. Yeah, what data do you use for this? Is it the CDISC data?
For this example, we used Pharmaverse Atom, but we've also used random CDISC data before.
Okay, perfect. Yeah, very cool. Awesome. Someone said if I missed a badge, yes, just send an email or reach out in the Zoom or excuse me, in the Slack platform. I'll put a link here to the Slack, and that would probably be the easiest place to get questions answered throughout this week because we're monitoring it. But feel free to post there. You can also just send us emails at info@rnpharma.com, so that should work too. We also posted the badges, so if you'd like to get a credential for today, fill out the form. Appreciate everyone for hanging in there for at least 75%. We'll post the recording on YouTube probably by the end of the year is our hope. And I think other than that, we're really excited to have other workshops kicking off this evening for APAC. And then there's a workshop tomorrow at 8 a.m. For the US Europe version of the conference. I think that's about it. Any other questions? Thank you so much. Really, really appreciate it. As always, I think I want to say at least your second or third workshop over the years. Maybe if Emily, I think it's at least your second one I know of. So
Yeah.
we always love having repeat instructors coming back and teaching us amazing things from the community. And with that, I think we can go ahead and close the bridge unless people have other questions they would want to tackle. It looks like one, actually, I think Daniel helped answer it in the chat box. So, all right. Well, I think we'll go ahead and close the bridge. Thanks again. See you, everybody.
Thank you so much.
Thanks for coming.